So I'm going to talk about multiplicities. I'm going to give my summary here, which essentially says that multiplicities dominate research. Tons of decisions are based on them. I'm about to tell you what they are. And my summary is that these are basically responsible for wasting 50% of resources in clinical trials because we make bad decisions based on them. And so you need to always interpret results in context, and you need to pre-specify your analyses. And I'll talk about why in a moment. This is going to be a bit more of an interactive talk. I'm going to be asking people questions and kind of expecting a little bit of participation. And I need a volunteer for the end who would like to claim to be an old statistician. Anybody want to be an old statistician? All right, Eric, you're an old statistician.
You go for samples.
All right, let's see. All right, so what's the definition of a multiplicity? If you're doing any kind of research, you can really get into multiplicities in a couple of complementary ways. So you can be doing an experiment and you're testing multiple hypotheses. So I may be interested in asking several questions within the same study. That could be considered a multiplicity. So, and again, the multiple here is the multiple different questions. The other thing you can do, which is kind of the complement of this, is you can actually test the same hypothesis multiple times. So whether this is doing multiple trials as part of a research program, or even within the same study, I'll talk about how you might do that in a moment. The prototypical example of a multiplicity is subgroup analyses. This basically happens all the time. People run a fairly large trial. The trial does not meet its primary endpoint, so technically this trial failed, depending on your viewpoint of failure. Remember, usually failed means doesn't answer the question, but it didn't meet its primary endpoint. Usually at this point, the investigator goes, well, I know this thing has to work. It's impossible that I actually did not get a positive result. So I'm going to go through and I'm going to figure out, surely it's just a matter of I didn't test it in the right people, or I didn't test it at the right time, or I didn't test it in the right dose, or something else. So they start snooping through their data and they go, let me just look at every possible subgroup. So I'll look at men, I'll look at women, I'll look at old people, I'll look at young people, I'll look at left-handed, right-handed, whether you have green hair. You know, have you ever seen these baseball statistics where they go, you know, so-and-so is batting 400 on Tuesdays when the weather's above 80 degrees. You know, it's the same sort of thing that's being done within a clinical trial. And you find out, oh, lo and behold, if I look at people over 80 who are not as sick, then the drug shows this enormous effect, and they report, you know, the p-value is 0.001. You don't know what they did here. They may have looked at the equivalent of 200 different analyses in order to get that 0.001. And then what they do is the following. Okay, now that I know who it works in, be careful of anybody who says they know anything on the basis of a clinical trial. Everything has a ton of uncertainty attached. You do learn a lot. You do not know much. But now that we know that who it works for, we go and run another trial, usually about the same size, that's in the subset. Because it's in the subset, it takes longer to run, and lo and behold, that trial fails. And this happens over and over and over and over and over and over again. And you actually don't see a lot of it if you just read press releases, because they will hype the subset result. Oh, I've now found it works in this subset. And then when the trial fails, it doesn't make any kind of failure. Kind of put that very somewhere underneath the side of that. So anyway, so what's going on here? Anytime you do a statistical test of any sort, there's a chance of getting a false positive. If nothing's going on, there's this 2.5–5% chance that you're going to make an error, just because you can never be. With statistics means never having to say you're certain. So you're going to run into this error possibility. And so even if nothing really works, if you look through lots and lots and lots of things, you look through lots of subsets, you're going to see a lot of random highs and random lows. And if you look at enough things, the highest of the random highs, they're going to be pretty darn high. And the lowest of the random lows are going to be pretty darn low. So you're going to find something. And just imagine you've got this 2.5–5% chance of making a mistake. You do this 10, 20 times, the odds are pretty high something's going to be there. You see this kind of effect with baseball players in, say, April. People are always batting 400, 450 in April. And that's because relatively limited sample size. We've got several hundred baseball players. Somebody is going to get lucky. It's the same kind of process that's going on here. So some subgroup gets lucky. When you then select it, there's nothing there. There's really nothing underlying. There's no fundamental truth here. So when you run the next trial, it has the 2.5 to 5% chance of making an error. And so you typically don't see anything and your results aren't confirmed. So you want to be really careful about this mechanism, that you're testing multiple things at once and what you know on the basis of those. So here's my first kind of quiz. Here's another situation. Simple design. I'm going to collect some data. When I get to 100 subjects, I'm going to look at it and go, is it significant? If I get a nice p-value, I'm going to publish it. Otherwise, what I'm going to do is I'm going to go, well, surely I'm not wrong. So I'm going to go and I'm going to collect up to 200 observations. Look again. And if I still don't find anything, I'll go to 300. As long as I get somebody to keep writing checks, this is great. So anyway, what's the problem with this? Yes, Victoria.
I think it's what we just talked about at the end of Chris's talk, right? The difference between power and effect size. The more people you add in, the greater chance you're going to have finding power, right?
Part of it.
Yeah.
So certainly the longer you go, the less you care about any results that you find.
But you have a better chance of getting a significant p-value, but the effect size might be the same, right?
Yes, and actually, let me rephrase that a little bit. It's worse. You're actually guaranteed, if you do this, you're guaranteed to eventually get a small p-value, even if the effect is 0. So if there's nothing going on, this is usually called sampling to a foregone conclusion. Even if there's just noise, you will eventually find something doing this. Now, you may have to wait a while for it. But this is, again, this notion, this is a multiplicity. I'm not testing multiple things. I don't have multiple subgroups. What I do have is multiple shots at the same hypothesis. So I can, again, get a significant result when there's really nothing going on there. So I have to be careful. Now, that said, I'm about to talk about this, there are designs that allow you to look at the data at 100, at 200, at 300, but you have to be careful how you do it. You can't just look for significance. Usually you have to, instead of using a 2.5% everywhere, you have to reduce those in order to compensate for the fact that you're testing multiple times. So, but again, this is an example of testing the same hypothesis in multiple occasions. There are subtle versions of this. So I do a lot of consulting in an area called historical borrowing, which basically means I'm about to, prototypical example, I do a lot of work in antibiotics. And people have a fair amount of idea about how antibiotics work. So if I'm comparing in, say, UTI, I'm comparing a novel antibiotic against doropenem, there have been clinical trials on doropenem. We have some idea how it works. About an 80%, 82% cure rate in multiple studies. So the question is, can I use that data in a future trial? It seems like I'm reinventing the wheel to do one-to-one randomization and enrolling a whole bunch more doropenem arms. If you think about what society has done, we've enrolled 100 Alzheimer's trials. We've done one-to-one randomization. We've got 100,000 Alzheimer's control patients. We probably know a lot about how placebos work in Alzheimer's patients at this point. So there are lots of subtleties involved in how to bring that data in, but that's the general problem. So that's kind of a side note, but that's why I was here. So a company comes to me and says, we've got historical data. It's from one of our trials. We ran a phase 2. This wasn't antibiotics. This was a rare disease. And what they wanted to do is they said, can we carry this phase 2 data? Can we bring it into phase 3? And so we talked, and it was in the same population, so they said, we're running the phase 3 in this kind of subjects. The phase 2 was on the same kind of subjects. Can you talk to us on how to bring that in? And so we proceeded to work through the design, the pros and cons of that. There are benefits and risks in doing historical borrowing, which I'm not going to discuss here. I did not ask the important question at this point. Anybody guess what the important question was, which I'm incredibly stupid not to have asked immediately? And I'm only stupid because I do this often enough, I should have known better. If you didn't ask the question, you're all in good shape. So the question I didn't ask them was, what other data did you look at? So they told me, here is our phase 2 data on the population we're about to go into in phase 3. So I finally asked them, how did you pick this population in phase 3? And they said, oh, that's not all our phase 2 data. We've got another triple that. And so what they did is they looked through all of their phase 2 data and did subgroup dredging and figured out which subgroup was going into phase 3. And so that process itself is prone to multiplicities. The population that they picked to go forward into phase 3, that's likely a random high. And so if they use that data and borrow from it, they're basically going to borrow off of biased data. And so that creates a major problem for this. So at this point, I said you can't do this, and I think we parted ways semi-amicably. They were disappointed. Anyway, yes, sir.
So, I mean, I understand your point, but still, if you are running phase 2, there is a chance, higher chance that you're actually targeting the wrong patient population, right? So how could you do it the right way?
So that's an excellent question. So certainly, and so I'm going to repeat your question just for volume here. So basically, this is a reasonable point. If we do phase 2, we often don't know the right patient population. It's often reasonable that it does only benefit a subset of the population. So how do we find that, and how do we take into account, we know it's prone to multiplicities, what do we do? That's a long talk to give you an answer. There are a couple things that can be done. One of them, actually, can I table that for a couple slides? So, but I am going to get there. There are a few things that you can do. Essentially, it means look, but look a little cynically. So you want to make sure that you feel more confident. Here's another situation that we run into. And suppose there's no data dredging whatsoever. So I work for a private consulting company. We work for lots and lots of companies. We design about 200 clinical trials a year. 150, 200 clinical trials a year. And if we're designing a phase 3, almost every one of them comes to us with good phase 2 data. And they go, oh, our drug's going to work. We did great in phase 2. The question is, should we take that at face value? And let's assume there's no data dredging whatsoever. So this is, they ran their phase 2, they ran it in the population they intended to, they didn't dredge their data, they are coming to us, and they do indeed have good phase 2 data. Do we take that at face value? There's no, they have not themselves, in theory, committed a multiplicity.
I guess there are a bunch of people who never came to you.
There you go. Perfect. Answer in the back. It's always, and by the way, I had a fantastic picture here that was the tip of an iceberg, and it showed the little part. And for some reason, I could not get the file to be less than 24 megs, and I figured Joy would kill me if I flew up her mailbox. So anyway, so I don't have the tip of the iceberg picture here, but it's worth remembering who didn't come to you. So that particular good phase 2 data, if 10 people out there ran trials, one of them may have found good phase 2 data. They're the people who are in front of you right now. Everybody else went, oh, my drug doesn't work, and failed. or didn't get funding, or whatever the case may be. So there's still a multiplicity there. And so we often, everybody goes, oh, this works, I got great phase 2 data. All the time we're like, we've seen a lot of great phase 2 data, and it does not pan out in phase 3. And that's because there's a general, everything gets progressed and gradually weeded out. That's actually one of the benefits of having different phases of development, is that everything has to get confirmed multiple times. But when you're reading, and remember, there's stuff coming out all the time on such and such works in mice. Ignoring the translation issue from mice to people, a lot of those trials simply fail because there are a lot of things being explored in mice right now. And everything that does well ends up being taken forward. But a lot of those are, again, the type 1 errors. We'll see you in a position to move me soon. All right, so now back to your question. What's your name, by the way? Ofer. All right, so back to Ofer's question. So how do you deal with multiplicities? If you know that they're existing, what should you do? And the hardest thing is to recognize they exist. So, and I gave you a couple more subtle versions. Always take everything into context. The analysis that's in front of me, what analyses are not in front of me right now that could have been done or were done and failed? It's always worth keeping that into account. It is very hard to quantify post hoc analyses. If somebody says, oh, we went through our data and we did analysis, that is a wonderful hypothesis generating procedure. It is very hard to quantify what the multiplicities were. How many analyses might you have done? Oh, I looked at this because I noticed that number was interesting. Could you have noticed something else? There's no way to get an idea on how many analyses are involved in that. So one of the things that you'll see, journals are very picky about this. All the analyses, you need to pre-specify them. And the reason for this is if you have a pre-specified plan, somebody can go back later and look at this and say, oh, here is what they intended to do. I have an idea about what kind of multiplicities are present in that analysis. And it's not perfect, but it certainly is a huge step forward. So this is why people want things to be pre-specified. There are a number of corrections. There are simple things you may have heard of, like the Bonferroni correction. If you want to test 5 things, then you have 2.5% one-sided type 1 error. You can split it up. So you use 0.5% for each of the hypotheses. And what this does is it minimizes the chance of you making any error. So if you have 5 hypotheses, your chance of making any mistake among the 5 remains 2.5%. It has some difficulties in that it's pretty conservative. If you want to actually test 100 things, then it's really hard to find anything doing a Bonferroni because everything has to be so significant. If you're doing situations where there are multiple interim analyses, I'm not going to discuss the details, but there are well-defined ways to do this. People have worked out if you test the first interim using this p-value and the second interim using this p-value, which is different, and the third interim using this p-value, which is different, then there's a way to control for 2.5% type 1 error. So consult your friendly statistician on how to do that, but you can get smaller sample sizes. You just need to do it right. There are gatekeeping approaches. So one difficulty with Bonferroni, for example, if I have 5 endpoints I want to test, but some of them are more important than others, well, I don't want to only have 0.5% on the one that I really care about. So a lot of times we employ gatekeeping strategies where you order your endpoints. So I might test the primary endpoint first. If that fails, then I'm done. The whole study, I can't claim anything significant. But if that wins, then I move on to my, say, key secondary endpoint. The advantage here, for technical reasons, is you can maintain all the type 1 error. Now, you've got to pass the gate, so to speak. But if you win the primary, you can retain your full alpha for your secondary, and then you can start to split it up in ways. You can actually make these graphs very complex and talk about 8, 10 endpoints and prioritize. And again, the notion is that I really want to make sure that I test my primary endpoint and the other things are not as important. Be careful how you pick the order. Roger has a good example of somebody, they had an endpoint that was relatively easy to win and another endpoint that they thought was more clinically relevant. They picked the more clinically relevant endpoint first and then lost, which allows them, they have technically a failed study because they lost that primary. If they had just done it in the opposite order, test the easy thing to win first, or what they perceived to be the easy thing to win, and then tested the clinically relevant endpoint, they'd at least have a successful study that then has some interpretation issues, rather than a failed study. This makes a difference on getting your paper accepted and everything else. There are other ways of doing this. People ran into problems when they did genetics. So if you're doing something, microarrays used to be the rage, you test thousands of genes at once. If you try to control the error rates for thousands of tests, it's really hard to do. So you need p-values less than 0.00001 or whatever the case may be. And people went, do I really need to protect myself against one mistake? If I make 100 decisions and 99% of them are right, is it bad that I made the one mistake? And usually the answer is no. If 99% of my claimed genes that are really doing something, if 99% of them are right, I'll accept the 1% that are wrong, even though I know I'm going to make those. So that's often, we use false discovery rates, where instead of saying, am I going to limit the chance of making any mistake? No, I'm going to limit the rate of the mistakes that I get over time. It's just a different way of thinking about how you're making mistakes here. All right, Eric, let me kind of do an example here to close up. Wow, look at that. I'm great with time. 5 minutes, that's where I thought I would be. 5 minutes, all right. And I've just been, people are tweeting about me. Great. Thank you, Michael. Anyway, I get that in real time on my phone now. Okay, so Eric, you are an old statistician. How many tests have you done in your life?
Thousands and thousands.
Okay, I'm good. Let's say 2,000 tests. Are you used up? Have we used up all your type 1 errors? There's a multiplicity there. Eric's tested a lot of things in his life. This is one reason my first day of undergrad linear regression, the teacher came in and said, "We're so glad you're here. We're all out of alpha. We need a whole new batch." And after 2.5% alpha, glad to go. Have you ever made a type 1 error?
I was going to say, my fiance would say I've got lots of errors.
I'm sure she doesn't consider herself one of them.
Sure.
Yeah, so surely he has. It's an interesting question on what is and what isn't a multiplicity. Usually we do not call statisticians, you have to consider every test that you've run in your life, and you have to have never made a type 1 error, so we need to adjust for that. Otherwise, it would be on our CV, have conducted 1,500 tests, remaining alpha 0.0000. So it would be on there, we hire on the basis of that. So usually we actually, if you think, we kind of go for false discovery rates. We are making, in the long run, about 2.5% type 1 errors, and we don't adjust because it is Eric Foster's 2001st test in that regard. Another place where we often don't adjust, if you've heard about master protocols, these are trials that test multiple therapies within the same trial. So you may be testing 5 different Alzheimer's drugs all at the same time. It's in the same experiment. We usually do not take a correction on that. There's actually a debate. If you go to Europe, they're actually a lot more. So Europe is actually, we don't know about that. The US is, no, we're definitely not taking a hit. One of the reasons that The arguments for not taking a hit there is I'm testing 5 things at once, I'm sharing a control, I'm more efficient. Marianne knows this well, I'm pretty experienced. In any case, I can easily run this as 5 separate trials. If I ran it as 5 separate trials, I would not take a correction. So if I put the trials together, I share operations, I share a control arm, I make things more efficient. The general notion is, why would I take an alpha hit on this? I again am working in kind of the false discovery rate notion that if I run this kind of process, I've got 2.5% overall. And that is subjective on what is and isn't. So if there's anything that I just want you to take out of this talk, pre-specify your analyses, specify how you're resulting, how you handle the multiplicity, and just be aware. Give some thought in everything you read. This is an analysis. What other analyses am I not seeing? And how do I interpret things in context? All right. Thank you all very much.
Any questions?
Yes, sir.
So I want to give you a scenario and ask you whether you would regard this as a form of multiplicity that's concerning. If you're interested, say, in the hypothesis that a certain drug improves functional outcome, and you have a primary and a secondary endpoint or a tertiary that are multiple tests of function in different ways. So you're looking at, I don't know, a timed up and go, or a 6-minute walk test, or a 20-minute walk test. Is that a form of multiplicity, or are you ensuring that you're perhaps seeing a consistency of effect to reassure you that any effect that you're seeing is actually sort of genuine or multiple outcomes that get at the same issue?
So I'm going to say yes.
I thought you might.
So it's certainly an interesting kind of multiplicity. So I've seen at an FDA panel meeting, company comes in with 8 secondary endpoints, and they're all measures of function like that. And they go, we want all of them. So surely we want 8 different tests. It is definitely a multiplicity in that it is multiple tests. So certainly that is happening. That is mitigated by the fact that those tests are likely strongly correlated to each other. If you win one, you're likely to win the other. If you have multiple tests and they're all 100% correlated, then it's not really a multiplicity. You're testing this, you're really just getting the same answer, and it's a cut and paste more than anything. So to the degree that they're correlated with each other, that mitigates the degree of multiplicity that's there. So, the example, if you have 8 things that are highly correlated, I view that as, you won 1 plus epsilon in terms of tests, and would correct on that basis. In interim analyses, if you do interim analyses very close to each other, those are highly correlated. So part of this aspect, I guess my quick answer is, when you're thinking about the magnitude of a multiplicity, the correlation has to be part of the correction of that, and that would be a technical issue to deal with.
Right, and I guess my question is whether you're giving yourself false reassurance by seeing an effect on multiple outcomes that you expect to be correlated, or is that real reassurance?
It is partial reassurance, I think. I mean, certainly, the other aspect, if you tested 9 things and they were partially overlapping, if you win 8 of them and lose 1, I'd still be reasonably, especially what the effect of the one you missed is. So I don't think, you don't want to confirming is nice, but you don't want to overhype highly correlated repeated results, because they're just the same thing over and over again. Yes, students first.
All right. So let's say this master protocol thing I thought was very interesting. It reminds me of preclinical pipelines for down selection. So let's say you have a dozen agents and you're running them all through the same animal model. These are animals too, I guess, right? And so if you were to get a positive result from 1 of 10 agents that you were testing in the same trial, maybe even with some overlapping controls. That seems an analogous situation to the master protocol that you raised. How would you address the multiplicity in that? Would you have to kind of take a hit on that a little bit?
So it's an interesting question. I would distinguish that situation where it's a company trying to trying to find something to progress in development, as opposed to the regulatory setting with the multiple master protocol. The company is certainly taking risks. If you test 12 duds, then you have a risk of finding a false positive. That said, early in development, you often accept a relatively high type 1 error risk as just standard to, 20, 30% is fairly common. We're only going to the next phase of development on there. The master protocol is a particular issue because the real question is not the risks and benefits for the company. The question is the risks and benefits. Does the company do better? Are they better being part of the master protocol and sharing resources? Or are they better by themselves where everybody's running separate trials? And I think the argument there is just the risks and benefits to society are the same, not an issue of, am I trying to make the risks small or big on that? So I distinguish those. In the preclinical, you want to know, how big a type 1 error do I have? The master protocol, the question is, if all the companies get together, do I put a higher bar on them? And that would just cause people to waste resources if you did. Chris?
This is a quick question. It's kind of the other end. I'm curious your thoughts because I've lost this battle numerous times. Where when you say pre-specified analyses, respectively, I absolutely agree with that. With clinical trials, inevitably, after it's over, there's a number of secondary post-hoc encounters, which are exploratory by nature. And more journals that we try to publish want you to adjust for multiple comparisons, which, in my opinion, so I'm curious quickly, in my opinion, if they're exploratory, and you say the word exploratory, I don't think it matters if you adjust or not. It's no more convincing one way or the other. I agree, you're just applying a false correction. I think you just answered the question, which is, but how do you approach that? I fought and lost that battle so much lately.
I don't think there's anything you can do. I report confidence intervals. If you pin me down, I'm a Bayesian. We don't correct anyway. So we have, our priors do. We use hierarchical models all the time, and it does do a partial correction to it. But there's nothing you can do. You give the confidence interval, and somebody else, if it's an exploratory analysis, I expect somebody else has to go test this. So go confirm. Okay. Right. Hey, Roger.
I think the question illustrates the fact that there's not really a one-size-fits-all answer to this. And the challenge over time is to get a better and better, deeper understanding of when multiplicities arise and what it is, and being clear about what level of assuredness you're trying to demonstrate with your statistical analysis. So I think that there's one of the reasons we often lose these arguments is people are looking for some sort of answer they can apply broadly across all kinds of heterogeneous situations, and it just life isn't that simple.
But getting your picture taken is quite simple.